our cancer vaccine, uh, Elpida 1. Imagine a world without cancer. This is our mission to provide prophylactic cancer vaccine. Cancer have many problems, but the main problem is the lack of cancer vaccine prevention method. Opportunity to our cancer vaccine, it's very high comparing with a classical vaccine solution. Impact of our vaccine solution, it's high comparing with existing treatment or vaccine in the world. Market strategy. Market strategy is designed in 3 steps. Step 1, production of vaccine, quality testing of vaccine and production of vaccine. Step 2 is testing safety and clinical trials 1-3. And step 3 is designed to sell vaccine and distribution. Solution is concept unique worldwide. Is consisting in administration in its concept unique worldwide, consisting in an administration of naturally occurring microorganisms. It has specificity against cancer tissue without affecting healthy tissue. Its cancer effect seems to be both locally against primary tumor and against metastatic tumor, secondary tumors. Side effect not have. It's highly efficiency and reducing the tumor or complete disappearance of tumor. Vaccine work as a treatment and as a prophylaxis method. Rules, uh, cancer treatment option, surgery, laser, chemotherapy, radiation therapy, target therapy, immunotherapy, hyperthermia, photodynamic therapy, stem cells transplant, blood product donation, and blood transfusion. Peri-tumoral injection, control fever, and outcome assessment. We studied 9 cases, mixed breed, masculine and female, different type of tumor, mammary, perianal, foot, etc., and with metastatic uh, cases. Our solution provide positive evolution of 75% from 4 cases in totally 9 cases of dogs analyzed. Number 2, it's a female, 10 years, mixed breed with mammary adenocarcinoma and metastasis of right legs, uh, posterior legs, and have a dimension of 2 per 2 per 1 centimeter diameter, and for the legs 5 for 4 for 3 centimeter diameter, and have a positive evolution of 1 per 1 per 0.5 centimeter, and the legs 3 for 3 per 2 centimeter. School age 1.5 years, mastocytoma, and size of tumor 2.5 per 2.2 and 2.1 centimeter, and with positive evolution and disappear of tumor in uh, 2 months. Variation of fever in rat study. Treatment induced fever irreversibly in 48 hours. You use five cohort of Winstar rats, and this show in experiment number 2 variation of fever. Statistics and success rate, very raw statistics. Success rate is 75%, reducing tumor, reducing metastatic, and no death. Apply potentially to more than one cancer type, breast, mastocystoma, other. Preliminary conclusion and outcome, very high success rate, 75%. Outcome, tumor absence, tumor reduction, absence reduction of metastasis, uh, with no major adverse effect, just middle or high fever controllable with common medication. Advantage of treatment and vaccine, potential advantage, high rate of positive outcome that existed treatment, cheaper, shorter treatment, do not require sophisticated equipment, highly applicable in various medical settings, no tissue toxicity or tissue accumulation of therapeutic agent. Mechanisms of action, potential mechanism suggested. Actions seem specific against tumor tissue without consequence against the surrounding health tissue. Consistent increased temperature suggests involvement of the immune response. Hypothesis treatment enhance the macrophage activation, allow efficient reaction against cancer cells. Next, I will present prophylactic cancer vaccine. The concept of Elpida 1 prophylactic cancer vaccine uh, is two component. Vaccine component A, consisting in a probiotic suspension of Lactobacillus acidophilus 60%, Lactobacillus casei 25%, and Bifidobacterium bifidum 50%. Dose use is 20 billion of bacteria per animal. And vaccine component B, murine tumoral cell suspension of RS1 hepatocholangioma. Dose it's 1.4 million tumor cells per animal. By reconstituting component A plus component B represent Elpida 1 vaccine. Animal experiment, materials and method. Materials and method, animals, experimental group, vaccine group, 10 Winstar rats, 5 masculine and 5 female, age 12 weeks, average body weight 140.8 gram. Control groups, tumor suspension of cholangiocarcinoma RS1, 10 Wistar rats, 5 masculine, 5 female, age 12 weeks, average body weight 150.7 gram. Procedure, extemporaneum preparation of the vaccine, component A plus component B, which will be administered to the experimental groups. Component A, probiotic, 2 capsules of Riflor Forte, 20 billion bacteria per animal in 2 milliliters saline per animal. And component B, tumor cell suspension of cholangiocarcinoma RS1, 1.4 million tumor cells per 1 milliliter saline per animal. The administration of the vaccine in animal with the experimental group and the tumor suspension in animals with control group subcutaneously in the left flank area 1 dose. Obtaining hepatic cancer in animal model. Hepatocholangioma transplantable rats model obtaining by administration of 2-acetylaminofluorescence in the food of Winstar albino rats. The tumor may be intrahepatically grafted or subcutaneously grafted. It develops slowly in approximately 30 days post-implant. We don't know metastatic in general, but invasive local level is produced progressive cachexia, the average of survival period and 80 days. The subcutaneously and intrahepatic grafted tumor are similar with regard to the growth in average, rhythmic, and histopathological appearance. The tumor parenchyma with extensive mucin, with clear fibrosis trauma, well defined by adjunctive tissue through a dense fibrosis capsule. Results and discussion. Evolution of vaccinated group, evolution of subcutaneous node vaccinated group, the oxidation of subcutaneous node expressed in a creamy, white, yellowish pus, odorless 7-day post-vaccination, and the resolution of image of subcutaneous node with post-oxidation ulcer, 11 days post-vaccination. 11 days post-vaccination, the spontaneous resolution trend of subcutaneous node was observed by oxidation expressing a creamy white yellow pus, odorless, by local ulcer and crusting with no suprainfection. The male revealed the most significant rates of spontaneous resolution, from 5 animals, and day 11. Evolution of control group. Evolution of subcutaneous tumor control group. In 26 days, diameter is 20 grams. In 35 days, diameter is 30 grams. In 43 days is 43 grams. And in 49 days is 46 grams. 28 days post-inoculation of tumor, all animal with the control group develop subcutaneous tumor, which grow progressively and which led to cachexia and deep alteration of the health condition, demanding their euthanasia. The tumor average diameter was slightly more in males than in females. Hematological profile in vaccinated versus control group. Evolution of a hematological profile in animal within a vaccinated group versus control group. 7 days post-vaccination, the females respond with a moderate increase in monocyte, relative monocytosis. In addition, both males and females revealed a relative neutrophilia compared with control, possibly due to the simulation of neutrophil mobilization to the injection site of the vaccine. 14 days post-vaccination, the monocytosis reported in the first 7 days in females was remitted. In contrast, relative neutrophilia maintained in both females and males compared to controls. Cytologic examination after 7 days of vaccination show result of cytologic examination of subcutaneous node in 7 days after the vaccination, and show neutrophilic inflammation, abundant neutrophil, mostly degenerate with nuclear lobes or hypersegmented vacuolated neutrophil, necrotic, amorphous material, cell lysis, free bacil or phagocytic neutrophil, probiotics, and colorated with May-Grunwald-Jimsa method. Conclusion. Our study demonstrates that the subcutaneous administration of Elpida 1 vaccine in Winstar albino healthy rats mainly trigger a stimulation of innate immune highlighted by a tendency of neutrophilia monocytosis as well as by the activation of phagocyte in neutrophil migrated subcutaneously at the injection site of the vaccine. Practically, a system phagocytic stimulation is not excluded, but of the circulating phagocyte migrated at the inoculation site and of phagocyte from the other organs, splenic, peritoneal, alveolar, etc. In addition to activating innate immune system, which is the first line of defense against pathogen, Elpida vaccine also seemed to have induced the activation of adaptive immunity by initiating of anti-inflammatory immune response, T helper 2, clinical materialized by full resolution of the subcutaneous node after 11 days from vaccination. The results of the clinical study are to be supplemented by specific immunological tests to clarify the local and systemic immunomodulatory effect induced by the subcutaneous administration of Elpida-1 vaccine in order to use as immunotherapy method for different types of cancer. Strategy for development of vaccine. What are you requesting? Secure IP for 20K, a control study in rodents assessing efficiency against primary tumor, dosage, survival, safety for 600K. Decision: go for human clinical trial safety phase 1 or go for vaccine development. Next, I will present traction model based on minimal success criteria of $10 million per year with pricing model of $200 per vaccine per month, expected customer lifetime of 2 years, and expected customer acquisition rate of 1%, need to attract 833 new clients in the next 3 years, and product market fit it's 420 clients. Competition. Competitors are a pharma producer of vaccine, research center, university, partners, retail center, distributor, importer, phytopharmacy service provider, and beneficiary it's patients with cancer and healthy patients for preventing cancer. Spectrum clients and motivation. Um, our clients suggest to use innovative vaccine solution. Um, Customers are who want to treat without side effect, with safety and friendly solution. And another type of clients predisposed to change to other type of solution. Potential to our vaccine is to grow 60% offline and 40% online sales. Scalability increased direct with distribution. Uh, Increase sales with direct or indirect channel. Strategy to develop vaccine for other type of cancer, and another type of grow and scalability with 20% each year. In the period of last year, a study published in the social platform um, with 800 participants was carried out regarding the need for the vaccine product, the treatment and prevention of cancer diseases. From 100 subjects of the study, 70% will go for this type of treatment of vaccine. Finance model. Finance model is B2B2C or B2B2G. Business model. Market size of vaccine in pharma, it's more than $10 billion per year. And our strategy is transform our limited in action society and listing of the Bucharest Stock Exchange, attracting new shareholders, sell of vaccine in new market with different channels and offline, online channels, increase our sales with 10% every year, and possibility to scale in international market. Potential to grow statistic potential to grow per year. Our potential to grow is by 20% per year, estimated for 10 million ton per year of vaccine, exporting with 3% annual production increase with 6 million ton of vaccine per year. The team's advisory board, professor of economy, doctor in science, Dr. Honoris Causa of Wallachia University, young Kukui with more 20 years of experience in finance and in finance area. Sabin Cincă, professor of Bucharest University, Scientific Research Grade 1, Head of Cancer Research of National Cancer Institute, Alexandru Trestoianu, Bucharest, Olga Vieru, Master of Science in Organic Food Product, University of Bacău, Romania, and Dan Iulian Santa, CEO, co-founder, Master in Science in Biotechnology, and Ileana Claudia Santa, economist and co-founder. The teams of collaborators, mentors, Zoița Diaconescu from UMF Cluj, Mariana Duță, UMF Bucharest, Sabin Cincă, National Cancer Institute Bucharest, Andrei Vodnar, USAMV Cluj. Summary, sincerely thank you for the team, for the mentors. Thank you very much.